The archetypal foraging behaviour of tropical seabirds is generally accepted to differ from that of their temperate and polar breeding counterparts, with tropical species exhibiting less predictable foraging behaviour associated with the less predictable prey of the tropical marine environment. Similarly, temperate and polar species have predictable annual breeding seasons, enabling them to profit during periods of the year when prey availability is highest, while tropical seabird species exhibit considerable variability in their breeding strategies. Until now, the reasons for such variation in breeding strategies between tropical seabirds have not been investigated. While some tropical species breed asynchronously, others adopt a seasonal breeding strategy. We hypothesize that tropical populations adopting a seasonal breeding strategy do so for the same reasons that temperate and polar species do. They have a predictable foraging strategy. Consequently, populations that breed seasonally exhibit more predictable foraging behavior than those that breed aseasonally and asynchronously. To test these predictions, over 5 years, Dr. Louise Soanes attached GPS trackers to two closely related tropical seabird species that colonize the same island, Dog Island of Anguilla in the Caribbean. These two species nonetheless exhibit markedly different breeding strategies. The asynchronously breeding brown booby, Sula leucogaster, and the seasonal breeding marsh booby, Sula dactylatra. The resulting data enabled us to examine the bird's foraging behavior. Loggers were retrieved from 172 brown boobies and 79 masked boobies, recording 770 brown booby foraging trips and 596 masked booby foraging trips. Our data suggests that at Dog Island, not only the breeding strategies but also the foraging strategies of brown and masked boobies have diverged. Brown boobies forage less predictably than masked boobies. How do our results suggest that brown boobies forage less predictably? The directions travelled from the colony out to sea during each breeding period were not uniformly distributed around the breeding colony within any breeding period for both brown boobies and masked boobies. However, while there was great variation between breeding periods for both species, there weren't apparent differences between the species in this regard. Yet, year on year, individual brown boobies had core foraging areas on average 2 to 3 times larger than those of masked boobies, suggesting that their foraging was less concentrated. Also, the level of core foraging area overlap between individuals from the same population in the same breeding period was higher for masked boobies, suggesting that the population of masked boobies used a more similar area to forage than did the population of brown boobies. The size of the core foraging area differed between breeding periods for both brown and masked boobies, but with greater variability in brown boobies. In summary, brown boobies have larger core foraging areas, lower levels of foraging area overlap between individuals, and greater variation in foraging behavior between breeding periods. Masked boobies adapt a more predictable style of foraging behavior, indicative of a more predictable prey resource. It appears that masked boobies have a foraging strategy more based on memory, visiting the same localized, seasonally predictable foraging areas and searching for prey frequently within a small area until they encounter a profitable prey patch. In contrast, brown boobies, which breed at any time of the year, exhibit more behavioral flexibility, presumably as they respond to the less predictable prey resources which vary in space between breeding periods, but may be associated with localized patches of lower sea surface temperatures which indicate ephemeral prey patches. Our results challenge the view that the foraging behaviour of tropical seabirds is always less predictable than that of seabirds breeding in temperate and polar regions, and highlight the considerable variability in the breeding and foraging strategies adopted by tropical seabirds, which demand further exploration. For further information about this study, you can read the associated peer-reviewed journal article, which is available on the website of the Journal of Avian Biology, or you can request a copy from the corresponding author.